from the MORFUN. So I'm Jean Franc ois Ghiglione. I'm working in the LOMIC, and I'm going to present you today a talk entitled Marine Microbial Ecotoxicology and Engineering. And this is this is part of the thing that we are doing in the LOMIC. And François will also tell you more about Biosensor, etc. later. So I will begin this talk with this nice picture. I always love to put this. It looks like a sky, and we cannot find the soft cross actually, but but when you look at the scale of this, it's the microorganism that we're looking under a microscope. And when I begin with microbial ecology, I came with really basic question. So the first one was, who are you? Talking about biodiversity, what are you doing? So it's the different function that we can find in the microorganism, and there are plenty. And with who, so really the ecology, the interaction between different organisms and their environment. So that were really basic question. And in terms of biodiversity, the main question we may ask is there an organization of the bacterial species in space? Because marine environment are so dynamic. We can ask this question about space, about time, about which scales, and what can influence the bacterial diversity in terms of environmental drivers of this bacterial diversity. So I'm going to give you some overview of what we did in the LOMIC, and then I will go directly to the wily ecotoxicology. But first I have to tell you a bit about that. So what we did really recently is sampling all over the world, and we did a lot of, it was a collaboration between 8 different countries, and we analyzed a lot of parasequencing, you know, this new technology about looking at the biodiversity. And we ended up with 800,000 sequences of 16S RNA, which describe the bacterial diversity. And with the different sample from the different poles, so from Antarctica to Arctic, and also in temperate and tropical waters. And what came out of this entire picture is that the bacterial diversity in the poles is way different from the temperate. This is the first driver of the bacterial diversity. And the next driver of the bacterial diversity is the difference between the ophotic zone and the mesopelagic zone. So that diversity in the ophotic zone is really different from the mesopelagic. So that was just a description of this. And one interesting thing when you think about biogeography is that we found really few common species between the poles, between the Arctic and Antarctic. But we found something like 14–15% of the bacterial diversity that can be shared from one pole to another. So it's quite few, but actually it means that the dispersion of bacteria can exist from one pole to another. So that was really a nice view of the diversity that confirm the theory of everything is everywhere, but the environment select the bacteria that are living everywhere. The other thing that we show was we couldn't find a latitudinal gradient of the richness, the number of species that are in the marine environment. We found almost always the same value of 2,000 species per liter, something like that, between even the pole or in temperate or whatever. So that brings together with this has been shown also for protists, for small protists, and that brings the bacteria together with this, showing that with the theory of Fenton and Finlay, that there is no latitudinal gradient of richness for little organisms. Okay, that was one view of the bacterial diversity all over the world. Let's go now to the second question about function, how much bacterial diversity can influence the biogeochemical cycle. And this is an amazing question, because you see such a small organism and you think about the big biogeochemical cycle. So you know this kind of picture about the linear trophic chain. This produces a lot of organic matter because of exudation or the dead animals. And all of these organic matters, for a long time we thought that the dissolved or the particulate organic matters sink in the sediment. But since the 70s or the 80s, from the last century, we know that the bacteria remineralize 20 to 50% of this organic matter. And this is why we are focusing so much in the bacteria. And you know also probably that the bacteria are grazed by ciliate, flagellate, and also viruses, and we call and because of the grazing, come back to the dissolved and particulate organic matter. So we call this the microbial loop. And this is something that we are really focusing in the LOMIC for a really long time. So the main result that we get by looking at this is that we found a direct relation between the diversity of the organism and this remineralize, okay, get it, of the organic matter in the water column. We also look really closely at the diversity and the activity of the bacteria that are living on particles. And we found really an underestimation of the role of the bacteria that are attached in the particle organic matter compared to what is known. And also in general, we found that the main drivers of the bacterial diversity, but also of the bacterial production, their activity, is the control by the resource, that it's much more important than the predation. So the bottom-up control is much more important than the top-down control. Okay, this was for a general view of what we do about function, and about ecology. It's more here the impact of the anthropogenic pressure on marine microbial ecology. And so there's two views when you think about anthropogenic pressure. Is there an impact on the microbial loop, but also what can be the services that microorganisms can give to help to understand the quality of the ecological quality of the marine systems. So this is what I'm going to talk today about marine microbial ecotoxicology and engineering. So in the LOMIC, in our group, the marine microbial ecotoxicology and engineering is one of the different teams, I mean, let's say teams, that main topics that we have in the LOMIC. So we have 4 different topics: the microbial processes and ocean biogeochemistry. So you can understand by what I said before, the way we look at the link between this small organism and the general global carbon cycle. There's also another direction in our team about the regulation of microbial function by light and nutrients. So François will talk about that later. There's another more focused axis in our team is the reactivity of organic matter, and really the link between microbial diversity and the degradation of organic matter. And the third group about marine microbial ecotoxicology and engineering that I'm going to talk now. So the first question you may ask about marine microbial ecotoxicology is what it is exactly. So the definition of this that I give, it's like, okay. It's the science working on the ecological consequences of anthropogenic pollutions at the level of microbial communities. Okay, it's kind of ecotoxicology adapted to microorganisms. So the way we look at it, we have 3 different questions in this. The first one is the effect of pollutants on the functioning of the microbial loop. So that's what we just discussed before. The adaptative and the resilience mechanism of microbial community and function under pollution. So that's really the impact of the pollution on microorganisms and the way they can adapt and they can come back to the origin before pollution. And also the identification of microbial genes, but also microbial species, really involved in the organic pollutant degradation. So I'm going to develop this 3 question, and also the microbial engineering. So what is the definition of this? Is really the science using the microbial resources. So the first one is the impact. The second one is really we try to use the microbial resources to answer demands of social, economical, and environmental demands. So that's really the service that are provided by microorganisms to help us, to know better the anthropogenic impact of pollutants. So I will go in the different question later. So the first is the effect of the impact of pollution on the microbial loop. And I focused from a long time, and in the lab it was like 20 years of research on petroleum. So this is one of the pollution we're looking at, but we're also looking at copper pollution and also pesticide. So, but one of the easiest one is the petroleum, because it's for the bacteria. You know it probably it's a carbon source. So it's like the same than here, homogenization of organic matter. I mean, hydrocarbon are organic matter that can be used by microorganisms. The thing is, when you add petroleum, it makes a disequilibrium between the carbons that is added in the environment, and but the supply of nutrients like nitrogen and phosphate that are no limited. So that create a disequilibrium that bacteria have to deal with, and also the toxicity of this kind of pollutants. So what we have made many different observations in situ in chronically polluted sites. So we have a really nice site in Marseille. There are plenty of refineries there, so it's quite easy to sample there and see the impact of the hydrocarbon compared to, let's say, more pristine sites. So we have this site in Fos-sur-Mer that is full of refineries, and this one is more pristine. So that's always like this. I mean, we go to chronically site and try to find a pristine site that is close. And so we work also in Mexico, in Terminus Lagoon. And also we began a collaboration with Rodrigo de la Iglesia like 3 years ago. With Laurent Quillet, that is a guy working in Rouen on the effect of copper in bacterial diversity, archaea, and function also. We also worked in microcosm, so like experiment under controlled condition. So we did that with different sites, with different, so chronically polluted versus pristine, with the addition of oil, nutrient, etc. I mean, the basic one. In Greece, we did that for sure in Banyuls, which is really pristine for us compared to the other environment, and also in Antarctic and subantarctic zones, to look at the effect of the temperature, for example, in this kind of process. And what came out of this is that when you have a bacterial diversity at the beginning, and you add petroleum, then the bacterial diversity changed quite rapidly. It decreased in richness. So this is really classical. And we know that the first hydrocarbons that are degraded are the alkane, because they are really easy to be eaten by bacteria. And this is really regulated by the nutrients. So we call it the bottom-up control. And you probably know that if you want to help an environment that is polluted by hydrocarbon, you just add nutrients, and then it helps a lot. So we did many different experiments with different temperature, different ratio between C and P, etc. And then we managed to increase the degradation of hydrocarbon by at least 10 times, which is not too bad. And so this is almost well known. We had some experiment on this, but what helps, I mean, our expertise was really helpful to look at the top-down control, because what happened when you add such a carbon source is that the bacteria are really growing, and after they are decreasing. And then we really looked at the effect of the flagellate and the viruses. And you can see that rapidly after the decrease of the bacteria, you have an increase that is related by the predators. And so we really look closely at these processes, and we managed to see that the top-down control is quite important, because you see that it's completely changed the abundance of the bacteria, but also that the viral lysis was much more important than the predation by the flagellate. It's 100 times more important than the flagellate predation. And what we really characterize is that, so when you add petroleum, you have a decrease of the richness, as I said before, and then you have some species that are really specialized to degrade the alkane, and they are really fast growing. And this is really typical for when you have a fast growing bacteria, then it decreases because of the viruses, and we call this theory virus kill the winner. because the winner here was the one that was eating the alkane. And then it just disappeared because the virus killed them. And then you have another community that appear that is less fast growing. I mean, they are growing really slowly, but they are able to degrade more recalcitrant hydrocarbons. So we're able to look at the different diversity, activity, etc. in different experiments. And really we showed that the microbial loop really drives the biodegradation of this kind of organic pollutants. So it's really important to look at not only the bacteria, because they are able to degrade, but also the predation that limits this degradation of the hydrocarbons. Another question was, okay, when you look at the diversity, it's quite indirect. I mean, you see a degradation of hydrocarbons, for example, and then you see a change in bacterial diversity. But it doesn't mean that these bacteria are really able to degrade the hydrocarbon. They can just survive and be tolerant. So to go deeper in this, we just use the technique. We didn't develop anything. This technique already exists, but we use it with so polyaromatic hydrocarbons that are the ones that are the more recalcitrant in the hydrocarbon. And we are able to make these compounds with stable isotope, so C13. And we take this compound, we add it with the bacteria, and the ones that are able to degrade this, they will be marked on the C13, okay? And the other one are normal. So in that way, we are able to look at the functional diversity of the bacteria that are able to degrade this kind of compound compared to the ones that are tolerant, okay? So we are able to do this in the different area, you know, in Marseille, so with the refineries, the pristine one, and also another pristine one in Banyuls. And when we add this kind of compounds, really recalcitrant, we always find, so it's, okay, it disappear, but never mind. We always find the one dominant bacteria, which is called the Cycloclasticus sp, and we can say that this kind of bacteria can be a bioindicator because it's really the dominant one when you add the pH. And this kind of bacteria, at the beginning of the experiment, when you look at in the environment, you can find it, but really few sequence. So it belongs to what we call the rare biosphere because so it's really they are really rare in the environment. But as soon as you put this kind of compound, they become really dominant because they are the only one able to degrade such recalcitrant molecules. So this was really important for the microbiologists because we think that the rare, they are not doing anything. I mean, if they are so poor in the environment, that means that they are not doing anything. But actually, when the carbon source change, then they can really be dominant. So we say that this rare biosphere may have a really important role in cleaning the polluted ecosystem. And one other interesting thing was to look at the tolerant bacteria that were able, I mean, they were not able to use this kind of hydrocarbon, but they are able to survive there. And we found that there was a really low diversity of this, and they are resistant to the toxicity, but they do not degrade it. And that was, so we found different genera that were able to survive like this. And in many other papers, they claim that these bacteria were able to degrade hydrocarbon, but actually they are not. They are just surviving. They don't care about the petroleum. Okay, and yeah, in the future, we're going to do metagenomic analysis on this kind of. I mean, this technique is really powerful because then you can go to functional diversity. And by doing metagenomic analysis, the main problem with metagenomic analysis is that you have thousands and thousands of genes, you know. But then it's a kind of screen. Thank you. You go directly to a community that have a special function, and then the metagenomic data decrease, and then you have a better view of the gene there. Okay. In France, so we try to develop this idea about microbial ecotoxicology because you probably never hear that. But so we try to build a project about making this growing. So we did the first meeting with 25 French laboratories, and what was interesting there is that it's not only marine system but also terrestrial and people that were working everywhere in lakes and whatever. So we define different way of where we should go in microbial ecotoxicology in France. And I will organize in May in Banyuls a meeting on microbial ecotoxicology. So the first one was really to decide what is going to be the main topics in the future, and this one is going to be more presentation for the people to meet together, but not only from marine but also from terrestrial, etc. So, and the idea is the same than here: is sharing as much tools as possible, because we have tools that are really adapted to marine, but some other developed tools that are really terrestrial or lake, but can be used in marine maybe. So okay, that's the challenge of this. Okay, I don't know the time, but I'm okay or not? Yeah? Ça fait combien de temps là, à peu près? Okay, cool. Okay, this part will be much shorter. So let's go now to the microbial engineering. The first thing is the bioremediation strategy to restore polluted sites. So when you have, I discussed a bit before about bioremediation strategies. Usually it's addition of nutrients, for example. This is the human that try to help the environment, you know. So that's what we call bioremediation strategy. So one thing on the pollutant, you can add nutrients, for example. And I just get a project funded on plastic. And the objective of this project is the characterization of microbial community that are associated to the colonization of the plastic, but also on the degradation of the plastics. Blah, 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 blah. Plastics, yeah. So it's really exciting project, and there are really few all over the world. Maybe you met Zettler. She came here, I think, a couple of months ago. And she was looking at the diversity of the microorganisms that are living on the colonization, but now the idea is to see how much we can help the bacteria that are degrading this kind of plastic. So we will develop a mesocosm experiment with different kind of plastic, like polyethylene, polypropylene, phthalate, etc. And we will look at the different processes like attachment, because so when you have a plastic, the bacteria will attach to the plastic and then develop a biofilm on it. And this is this biofilm that exudate ectoenzyme, like lipase or things like that. And then you have that makes the erosion of these plastics, and then the degradation of the plastic can begin. But it's really low process. I mean, it takes really long time. For plastic, for this kind of bottles, I mean, it can stay more than 100 years in the marine waters before being degraded. So we are not going to work on the bottles directly. We're going to work on small pieces less than 5 millimeter. And the idea also is to make them grow on this plastic. So these colonies will be able, I mean, this biofilm will be able to grow on it and be used to it. And then after to add, again, the stable isotope, for example, polyethylene, and use the stable isotope probing, and to characterize the bacteria that are really degrading this kind of plastics. So again, this tool, we're going to use it again here. Okay, the second one was the ecological quality of coastal environment. So microorganisms can help a lot in this by bioassay, bioindicator, and biocaptures. So we're using different tools for the monitoring of marine pollution. There's a European project that are developed on this and to characterize the quality of the environmental marine systems that probably exist in Chile. And we have different tools that are using organisms. So the first one are the bioassay. So the idea is you take the water in the environment, you bring it back to the lab, you put it with certain organism, and you see if they are dead or not because of the pollutants. So there are different tests like Daphnia. We are using the microtox because it's a Vibrio fishery, so it's a microorganism, and we are using it a lot in the lab. So this is one part, bioassay. The other one, and all of them are necessary to really build an idea of the quality, the ecological quality of the system. The other one is the bioindicator. So here you go directly in the environment and you look at the different species that are there. And depending of the species that are there, you know that some species are really doesn't like the pollutants, and some others don't care too much. So most of the time, this study was done by another team in Banyuls, the LECOB, and they are dealing with the macrofauna. And the macrofauna can be used as an indicator of the quality of the system. Okay. And the one that we are trying to develop in the LOMIC, so it's really the beginning for us on this, but we are trying to go into this, and François will give a talk, an entire talk on this, so I'm not going to talk too much about that. But the idea is to use molecular, biochemical, cellular, or physiological characteristic, and then see how much. So this is much more. How do you say this? This is really more on one molecule, for example. So you want to focus on the. Okay, I'm explaining after. So how do you develop a new biosensor? We are dealing a lot with luminescence biosensor. So I was working a lot with this bacteria that is Marinobacter hydrocarbonoclasticus, hydrocarbon, so hydrocarbonoclasticus. And we have the entire genome of this. We develop chips on it. And then by knowing this, the different gene, you can do mutants on this. So the idea is, for example, I was really interested by the hexadecan, which is a really long alkane. And we knew where it was more or less in the genome, so we can do genetic engineering, and then you can change the gene that is usually used for the hexadecan degradation, and then you do mutants for that are luminescent, and you keep the promoter here, and then you have the expression of light when the hexadecan is present in the environment. So this is what we call a biosensor. So we have a lot of mutants of this. So we have only 2 mutants out of the 4,000 mutants. We have 4,000 genes, more or less, there. We managed to find 2 mutants of the hexadecan, so it's a new biosensor. And with this kind of tools, I mean, you can do it with many different organisms. So, and François will talk about the way he managed to build a new biosensor with Ostrococcus tori. So I'm not talking too much about that now. But the idea, what I would like to go is, you know, one organism can have his own sensibility to one type of pollutant. Okay, so it's important to have many different organisms, many different model organisms that have different sensibility to the environment, but also to the pollutants that you want to go in. So we can target general toxicity, but also the idea with the biosensor is to check different, so the alkane, the hexadecane, the naphthalene, for example, use different biosensor. Ostrococcus tori, it can be used for the pesticide. Also, we'll talk about that later. So the idea is to build for the LOMIC a platform of luminescent biosensor that can be used to help to know what kind of pollutants are there and how much they are able to be available for the microorganism. Okay, I had this European project that is finishing this year called Vermeil Ecotox. And the idea there was really to put all together these different tools. So, for example, here we were working on station d'épuration, wastewater treatment and harbors. And you know, when you have a lot of water, the river is really huge. So, les crues, comment on dit ça? Floods. Okay, thank you. So, different conditions. And then the idea is first to look at to do chemical analysis. So we're not doing this. We are doing this in collaboration with other people. So I call it exposition of pollutants. And we are also looking at different pathogens there. And the idea after is to give an idea of the danger. So meaning that you take the water and bring it to the lab, and then you use the different tools. So the different tools we are using is, so the macrotox, it's just the idea is to see the development of the organism, or if they are dead, just dead, you know, because of the sample you are bringing back to the lab. So we have different one. So that's the one I'm using more or less, and there's other team in our lab that are using the embryon development of sea urchin, Amphioxus, sea wolf, for example. So the idea of Biosensor platform is here, and with the same sample. And also we have colleagues that are doing the macro fauna and the meiofauna, you know, to have the bioindicators of the environment. And in the same project, we are looking at the bioaccumulation of the pollutants in different organisms, so in bivalves and fish. And then the idea is to bring all the things together, see how much they are sensitive, because it's not a really polluted environment. We find some pollution, but not so much. And it's usually the problem with pollutants, it's a mix of different pollutants. So the organism will answer differently. And the idea is to bring all the things together and try to find some standards, indexes, use some indexes, and find one for our region. Okay, I'm on time or not? Yes? Okay. So this, I'm not going to talk about that, because François will talk about that. The idea also with Ostrococcus is to produce a substance of interest. So you will talk about that. I'm not. Yeah? Okay. So, for example, there's okay. Okay. If I'm saying wrong things, yeah, just correct me. But the idea is, for example, you know the fatty acid polyunsaturated omega-3. So the idea is to use this organism and all the genetic transformation that François teams know how to deal with, and you can stop one metabolic pathway and then try to produce such kind of substance of interest. So if you have questions about that, go to François. And the last one is just an example of the different things that we are doing in the LOMIC. So I also work on accelerated biocorrosion of carbon steel. And this is a topic that I really love because it's really ecological microbiology. And the idea is that the carbon steel, so you find it everywhere in different arbors, etc. And there's an accelerated corrosion. And the physician came to us and said, okay, this may be a biological process behind this. And finally, we found that in the low tidal zone, you find this accelerated corrosion. And we find different microbial processes. And because of the exchange of electron, you know, they accelerate the degradation of the carbon steel. So we characterize different, so we made a little model of the biofilm that is associated to this biofilm with the sulfate reduction, you know, that is linked with the iron cycle, etc. So this is one thing that we were doing. And also I have a project now with the accelerated biocorrosion of stainless steel. So the same thing, I mean, we are trying to characterize as much as we can the microorganisms that are involved in such process. Okay, I'm done. Thank you very much. Yeah.
Francois, usually when the people who are working with the models ask the science